What was that?
Oh, just, I mean, you're going to be moderating this, right?
Yeah, so Isabel, we'll do a quick intro. I'll do a quick recap of the previous two meetings and then introduce you and pass it off to you.
All right. Welcome, everyone, to Renegade Research Roundtable. If this is your first time with us, thank you for joining us. We are glad to have you. I'm Isabel, CEO of our organization. We are a patient and caregiver-led, decentralized 501c3 nonprofit organization focusing on infection-associated complex chronic illnesses, such as MECFS and long COVID. With this, we have 3 areas of focus. We focus on research that's agile and patient-centered, education to increase the number of clinicians treating us, and community, where we focus on practical advocacy around barriers to care. Our work is made possible by our wonderful team of volunteers who move the wheels of our organization. Most of us have personal interest in advancing research and care of these conditions. To stay in touch with us and hear about upcoming events, please subscribe to our newsletter and follow us on social media. We will be posting the links for you to have handy shortly. Our work also depends on donations of all sizes. You can help us by donating to our funding campaign and also by sharing our campaign with others who can donate. We have more great and informative roundtables coming up, so please visit the event page of our website, renegade-research.org. And now I will pass it off to Tess to get us started with today's programming.
All right. Thank you, Isabel. I'm Tess, founder of Renegade Research. And I just have to say that I'm so excited that all of you came today. I was telling Robert and Rob that when I first started digging into the literature of MECFS, I tweeted that I really wish that I could be in the same room as Robert Naviaux and Rob Fair. And here I am with so many other people. So I'm very excited today. I'm just going to do a very quick recap of the previous 2 meetings in this series to kind of set the stage for people who weren't here for those. So this started off as it came out of Ron Davis's meeting last fall, and I saw Ron and Jen are here, and some people from our team were there, and we knew a bit about the mechanical basis model. Jeff Wood is here, who came up with this, and we know a lot of patients were finding that they had mechanical issues, but then there's all this other research in MECFS showing abnormalities at the molecular level. And so we wanted to know, how does all of this connect? And that's how this meeting came to be. So we started off with the first in the series was related to the brainstem and the specifics of these mechanical conditions. So Michael Van Elsker talked about the brainstem and how it has sickness circuitry, how it responds to infections and signals from the vagus, and mentioned that those signals don't have to be infectious. It controls systems found to be dysregulated in MECFS, and there are imaging abnormalities found in MECFS in the brainstem. So you can go to our YouTube channel and watch this meeting. I think it's really worth watching. Van Elseker was there. Jeff Wood talked about like a sort of an overview of the model. And then in part 2, Herbert Renz-Polster added to this list of things that can come from the brainstem, the stress response. So if there are issues in the brainstem, the stress response can be affected. And then we also, in part 2, had some results of MRI imaging of the brainstem. We also dug into some kind of specifics of what these mechanical conditions are and how they could cause deformation of the brainstem and how the incidence might be high in MECFS. And I think we have some people from this paper who are here today showing these high numbers of incidence of these conditions in MECFS. There are anecdotes of patients who have gone into complete remission from addressing these conditions, and I think some of these patients are also here today. And then Dr. Yelman in the first part talked about the clinical perspective and how they are seen clinically and how they can improve with treatment, but that they aren't the only part of the picture. It's a complicated condition. So like I mentioned, originally in Ron's meeting, we wondered could these possibly connect to downstream effects? And I mentioned the lack of the stress response, which Herbert has hypothesized could lead to post-exertional malaise. That's in the part 2 recording. We had some lightning talks as part of that, and Dr. Houghton brought up possibility of hypoxia and how Dr. Bluestein, who I think is also here today, talked about mast cell activation. And then today, Dr. Naviaux will be talking about the cell danger response and how this all kind of connects a lot of the pieces. I put a little star here that some of these could be upstream or there might be feedback loops because, again, this is a very complex condition and there's a lot of things going on that could feedback. So it's almost even hard to say what's causal in this case, I think. So today we'll have Dr. Naviaux talk for about 30 minutes and then we'll open it up for Q&A and discussion. For those of you who have been to the previous 2, you know that this can go very long. They've lasted for up to 3 hours because we just keep going as long as there's good conversation happening, but feel free to drop off at any point, and this recording will also be posted on our YouTube channel. I just wrote down some possible discussion topics. We'll see where the conversation goes, but there are a lot of people here who study different parts of the system, so maybe people have ideas for other connections here. Maybe talk about how to evaluate some of these hypotheses related to the mechanical conditions and these connections. And then last time, Athena Hall, who's also here, talked about ideas for like creating a global consortium or how we can move forward discussing these issues. Okay, so that was the quick recap. And now I'll introduce Dr. Naviaux. So he's a professor of genetics at UCSD and directs a core lab for metabolomics. He's the co-founder and former president of the Mitochondrial Medicine Society and a founding associate editor of the journal Mitochondrial. He's a discoverer of the cause of Alpers syndrome, the oldest Mendelian form of mitochondrial disease, and the developer of the first diagnostic DNA test for it. He's a Salk-trained virologist and molecular and cell biologist and was trained at NIH in tumor immunology and natural killer cell biology. His work in ecosystem dynamics has guided new work in microbiome ecology and metabolism in autism spectrum disorders. And he is one of my scientific heroes. Some of his papers on the metabolism of MECFS and cell danger response were what really opened my mind when I was reading the literature. And so I'm very excited to have you here today, Dr. Naviaux. And so I'll pass it off to you to share your slides.
Thank you very much, Tess. Let's see, I'll go to my slides. And see, can everyone see this slide here, the title slide?
Yep, looks good.
Okay. So my thanks to Tess and Isabel for reaching out and inviting me to talk today. I'll speak for about 30 minutes. About the first third of the talk deals more directly with kind of brain mechanotransduction, the process at which mechanical deformations can lead to molecular responses. And then I'll give a perspective about the CDR and ATP signaling. Okay. So these are my disclosures, and I'll start with this statement about pathogenesis of CCI, and let's see if I can, so MECFS shifts metabolism into this long-term hypometabolic persistent state that's we liken to a phenomenon of persistence called Dower. In broad terms, it's an evolutionary response that's enabled our ancestors to survive hard winters, pandemics, and famines by decreasing the amount of dependence of cells on neighboring resources. But MECFS persistence also, results in this physiologic increase in stress-related collagen turnover, and we got our first hints of that from our metabolomics paper on MECFS in 2016, where hydroxyproline and pyrroline 5-carboxylate, which can be synthesized from proline, were both elevated. And so these are molecules that can derive from collagen turnover. And if MECFS persists for a long time, then it can lead to this acquired form of connective tissue disease that of joint laxity and hypermobility that has become known as EDS3, and so part of this we would see as a result of CDR activation of inflammation and mast cell release of chymase, which is an elastase, and tryptase, which is a collagenase that weakens ligaments that support the head and neck and other connective tissues. Okay. And there are case reports of chronic infections with Bartonella that lead to inflammation and EDS-like symptoms that have improved with the eradication of the chronic infection. I put the PubMed ID here that you can look at. And then in patients with MECFS and documented CCI, surgical intervention can help remove what we would consider one of the CDR triggers that might be the most relevant one for a given patient. But oftentimes there are other cell danger response triggers that still remain that can cause symptoms to drift back. Okay. I apologize for the poor quality of this. I just pulled this off the internet, but it's basically, the slide is to begin the discussion of mechanotransduction, which is the mechanisms by which anatomical deformations or mechanical deformations in cells can result in the activation of the cell danger response. So just to orient people, so here's the pons, here's the cerebellum. Just inferior to the pons and at the lower edge of the fourth ventricle are the vagal nuclei. And here we have bones of the skull, the clivus, first cervical vertebrae, and here's the odontoid process. But all these things, when the brain, the skull is moving, the anatomical pressure on these sites can lead to stress responses that can influence overall vagal signaling and other signals. And you just have to, it's important to keep asking yourself, if a cell is anatomically deformed, how does it tell other neighboring cells that it's being stressed? Well, the answer is through released compounds. Okay, it releases signaling molecules that pass on the message. Okay. And so here's one example of that mechanotransduction done showing osteoblasts in culture that have been exposed to just culture fluid shear stress. And here there's, these are osteoblasts that are normally responding with actin. Yes? Did someone say something? Okay, so and audio is still on, so people can still hear me.
Yeah, we can hear you.
Okay, good. So you can see these actin filaments and collagen is stimulated from these osteoblasts. But when you pre-treat these cells with ATP, it inhibits their ability to respond to that stress. And ultimately, the normal response is to release some ATP. It produces some reactive oxygen. It activates some microtubule-associated protein kinases like the ERK kinases, NF-kappa B, and that ultimately inhibits maturation. If you look at other aspects of this, you can study, actually how the heart is, deformed with every contraction. It stretches and contracts, and actually that results in ATP release as well through these connexin 43 channels. What that also is associated with is depolarization. So depolarization, itself will release ATP. In addition to ATP being involved in depolarization. But in addition, this depolarization event releases intracellular calcium. And you can see over here this highly fluorescent, fourth figure on here is fluorescing with calcium. This is an illustration of how shear stress leads to ATP release from the cell that then binds to, in this case, P2X4 on these specific ionotropic forms of ATP receptors, and ultimately that leads to calcium release. Depending on the cell type, it can come from ryanodine receptors. It can come from IP3 gated receptors in the endoplasmic reticulum. But ultimately, calcium becomes involved after ATP signaling. So it turns out that just last year, a group published work on how tenocytes, these tendon-producing cells or tendon-associated cells that produce collagen, can be their signaling abnormalities can be improved by the treatment with suramin, okay, which inhibits abnormal ATP-stimulated CDR signaling by being a competitive inhibitor of ATP signaling at over half of the 19 different purinergic receptors that are known. So just to quickly go through this answer to the question, what is the cell danger response? Well, it's actually governed by thousands of genes. And, Ron Tompkins actually identified many of those in his, severe trauma studies that he took care of as a physician, and then analyzed the genes involved in the response. And then we find that, these are involved in about 30 different key metabolic pathways. And the CDR has 3 choreographed phases that have to actually be, used in sequence in order to return in order to initiate the healing response and to recover after any threat. And if you go to our website, you can click around and learn a little bit more about the healing cycle. But ultimately, it comes down to the role of mitochondria in the cell. And the really important thing for understanding, like making sense of this, is the conservation of matter and energy. Okay, and so the CDR is biologically expensive. And because it's energy and resource consuming, recovery cannot be completed until the CDR has been completed. Okay, now let's step back for a little bit and take this bigger question. So many of you, if you read through our papers, know that we draw a line between acute injuries and illness and the treatment of those acute injuries and the recovery after those injuries. Okay, and so on the left, you can imagine a whole system, in this case, a lighted house that can become threatened by infection, trauma, toxins, fear, pain. And physicians have been trained to, do a good job with managing these acute sources of damage, whether it's antibiotics, antivirals, statins or insulin. But what we find afterwards is that, once the acute illness is treated, we're left with cells that need to be repaired. And for, essentially 5,000 years of modern medicine, physicians have been taking, credit to some extent for, this repair, when in fact, what's happening is that a highly conserved sequence of events has been activated once the damage has been contained or eliminated. And ultimately, it results in cells having to create new resources and allocate energy in different ways in order to repair that that damage. And that's a process that we call the Salugenesis. It comes from the Roman goddess of healing and personal welfare and health. And it's an active process. It requires energy and matter. And obviously, if that energy and matter is being, consumed on one side of things, it can't be used for healing. Okay, and that's what we need to do. So this is the step that we believe is most, important for most chronic illnesses. Is yes, we have to get rid of the acute problems. Okay, the things that are causing the constant or repetitive stress. Okay. But then we also have to unblock the pathways that are involved in recovery. All right. So that leads to this view of human health that is bidirectional. And for most of most of modern medicine, or most of medicine period, has really been dominated by this idea that health can be interrupted by a number of different environmental factors. It can be, or genes too, but pollution, toxic metals, microbiome changes, infection, that lead to a state of injury. Okay, and so these things are acting upon cells to produce a state of injury. Okay, and that's a process by which health converts to disease that has been just called pathogenesis. But what I'd like to really focus on is that once pathogenesis has occurred, and you have eliminated the factors that might keep injuring the cells, then the cell has to actually get from this state of compromised function back to a state of health. And that turns out to take, it has a beginning and a middle and an end. A beginning, middle, and end. And the mitochondria that are associated with this are changing. So that many of you will know the M1, M0, M2 nomenclature is used for macrophages and microglia. It means pro-inflammatory, kind of undifferentiated for M0 and M2 are the anti-inflammatory cells, but also those cells have those phenotypes because of the mitochondrial states within those cells that are maintained. Now, it turns out that environmental factors in our exposome or re-injury, for example, you sprain an ankle and re-injure it 6 weeks later, it takes less force to injure it, will inhibit this process of return to health. But I want to emphasize that this green line back to health is not the reverse of the black line that led you to disease. It simply eliminating any one of the factors that led to disease does not automatically turn you back to health. There's an active process called salugenesis that has to occur. And ultimately, at each step, it's maintained by ATP signaling that will, if the cells continue to release ATP and it's not metabolized, it'll inhibit progression along this salugenesis pathway. Okay, there's age. I won't go into the aging aspects of this. So here's this, kind of alternative way of seeing chronic illness is that we believe that 90% of chronic illness relates to abnormalities in the process of recovery, of the molecular steps and mitochondrial transitions that are needed in order to drive, to repair and to drive recovery. And so, so what we really would like is to develop salugenesis therapies. And why don't we have these? Well, because we didn't know how to look. So, so it turns out that scientists are not very good at studying something if they don't have a name for it. And that was a big motivation for providing this term salugenesis. If you want to learn more about it, you can, and you can look up, our work on this. There's some nice figures that can help with understanding different aspects of this process, which is very complex. All right, so if you distill this down, what we ultimately come to is, what is the most fundamental measure of cell danger? And and that's extracellular ATP. Now, I will point out for those who are interested in reactive oxygen species, hydrogen peroxide, superoxide, hydroxyl radical, et cetera, that we cannot actually separate extracellular ATP signaling from ROS. In every case it happens, ROS is also produced, okay? The difference is that using antioxidants doesn't stop the signaling, but using antipurinergic agents that inhibit ATP signaling does stop the signaling. Okay, so for those of you who may not know, is the adenosine triphosphate, or ATP, is it's a purine. So that's where the word for purinergic signaling comes from, and the purine just has to do with this double ring structure over on the right, and it's the most expensive chemical that cells make. Okay, and ATP is more universal to cell life on the planet than any gene. Inside the cell, intracellular ATP is an energy carrier and participates in hundreds of chemical reactions. So in that case, it's matter. But outside the cell, it takes on its alternative function as a signaling molecule where it becomes information. And so the receptors that actually allow a cell to respond to extracellular ATP are on every cell, and that ATP signaling is more universal than any neurotransmitter or any cytokine system. So more than dopamine, serotonin, GABA, glutamate, acetylcholine, et cetera. Okay, so what does ATP do? How is ATP what does it do to inflammatory cells? Well, here's a plate of microglial cells that into which, whoops, let's see, we're missing something here. Oh yeah, okay. So we can put a drop of ATP in the middle of these cells in the plate, and then just photograph them in time lapse and look at what happens. So it turns out they send out pseudopodia because ATP is chemoattractant. It basically signals that this is an area where there's been cell injury. Except in this case, there was no injury. It was just the ATP itself that was added experimentally. Okay, ATP is chemoattractive for neutrophils, macrophages, and T cells. And you can block this with a variety of different antipurinergic drugs, including suramin. Okay, what does ATP do directly to mitochondria? Well, it hyperpolarizes them. It actually decreases their ability to make further ATP, and so ATP, extracellular ATP is active physiologic suppression of mitochondria. Okay, and what we found in patients is that over months of time, they become hypersensitive to smaller and smaller amounts of extracellular ATP, and that we can reestablish that balance by desensitizing the signaling pathway with antipurinergic drugs to help restore resilient health and, in principle, decrease PEMs and CRASs in MECFS. So this is another picture of the regulation. All cells release graded amounts of ATP in response to stress. These are the channels that will open up under stress conditions. They include, thiol residues that can be oxidized under oxidizing conditions that further open up this channel. Imagine a water balloon with pinholes in it that is placed under pressure. The more pressure the balloon is under, the greater the plumes of water released, and that's what's happening with ATP. You can imagine maybe that's a more direct analogous to mechanotransduction, but it's more complicated than that because the membranes around the subunits of the channels actually change by increasing the amount of cholesterol and ceramide and sphingomyelin. They become the oligomers, the monomers that make up channels are brought together in membrane lipid rafts that are stabilized by the lipids that can stiffen the patches in the membrane. Okay, so mitochondria are constantly talking to the nucleus through what we call the short grade retrograde path, and then also mitochondrial metabolism through ATP, and it turns out alpha ketoglutarate, succinate, a variety of different molecules, ketone bodies too, okay, regulated by mitochondria, are involved in what we call a long path retrograde signal back to the cells that alters gene expression. When these channels open up in the membrane, it results in a dissipative loss of ATP. You can call that purinosis. And it saps the energy of the cells releasing the ATP because it reduces the amount of ATP within the cell that's needed for healing and recovery. Okay. So anyway, when looking for drugs, that could block the loss of that, and that could also inhibit the signaling through these purinergic receptors. And it turned out at the time, the only drug, and even today, I mean, so this goes back to 2010 when we did some of the first searches. The only drug available for human use was Suramin, okay, which had been used to treat African sleeping sickness caused by trypanosome infections in Africa for almost 100 years. The pannexin channel is another target that releases ATP and can be inhibited by pannexin-targeted drugs, and there's companies that are dealing with that as well. Now, I wanted to know, okay, well, if this is a signaling molecule, if we inject it into animals, we should be able to actually measure volatile organic compounds in the breath of those animals, and then also do metabolomic analysis, acutely and subacutely at 4 hours, and get a better picture of what's happening. And the people that helped with this are Zuela, my wife Jane, and a very talented Japanese neuroscientist named Tomohiro Akiyama. Okay, so we injected the mice with ATP, put them in a little bell jar, and sampled air that they produced. And what we found is that they produced many different kinds of molecules that were small carbon and sulfur containing molecules, including carbon monoxide, methanol, methane, but also acetaldehyde, which is normally a product of ethanol. Okay, so we didn't detect ethanol in the breath, but the product of ethanol, acetaldehyde, was increased. And you'll hear stories about how sometimes, well, I won't go into it. So, but acetaldehyde, acetone, butyraldehyde, and this intermediate of cholesterol synthesis called isoprene was also increased. But cytokines and chemokines like CXCL1, IL-10, IL-6, and corticosterone were also increased. And when we looked at metabolism, we had many compounds that were increased, including dopamine that has nothing to do with purines, in addition to purine products like inosine and hypoxanthine. And what we found is that ATP injection produced more changes in the metabolome than any drug we have ever studied. And the only thing that's ever come close to it is lipopolysaccharide, which actually also produces ATP and produces many of its broader effects, non-TLR related effects via ATP release in the cell. Okay, well, what happens to the actual mice? And if you look at their oxygen consumption and you inject ATP, what you find is that it instantly created a state of fatigue, so that their mitochondrial oxygen consumption dropped by 74%. That's the initial cell danger response. And then there's a recovery response that is characterized by actually the respiratory exchange ratio, which is the CO2 produced over the oxygen consumed, actually falls during recovery. And that is because after the ATP is metabolized, to recover from it, there actually has to be a rebound activation of mitochondrial fatty acid oxidation. That turns out to be important. We can measure fatty acid oxidation in exercise ergometry studies. And it turns out in these studies that I've referenced down here, that if you measure carbohydrate oxidation, it is continuously increasing under stress. So glucose is used more and more and more and more, and fatty acids are used less and less and less. And that it turns out that the release of ATP from the stressed cells is actually blocking fatty acid oxidation in mitochondria, and stimulating and participating in the production of malonyl-CoA, which is a direct inhibitor of acylcarnitine delivery to cells. Yeah, so key exhaustion. Okay, so that's exercise to exhaustion produces this separation between carbohydrate metabolism and fatty acid oxidation metabolism. Now, this group also looked at patients with long COVID, and it turns out that if you measure fatty acid oxidation on the left side and look at the long COVID patients and put them under load, they will drop to exhaustion very, very quickly. Controls had an IC50 of about 195 watts on the bicycle ergometer, and endurance athletes could generate up to 320 watts. Now, this interesting hypothesis was that performance is proportional to the ability to handle extracellular ATP signaling or resistance to accumulation of ATP. And then on the other hand, your fatigue is associated with proportional to extracellular ATP sensitivity. So one hypothesis is that anticholinergic drugs like Suramin could inhibit this fatigue caused by ATP. But a caution is that these anticholinergic drugs like Suramin may be a performance-enhancing drug concern in athletes if this is actually a true hypothesis. I won't go into that. So the conclusion, so we're wrapping up. One is the two primary functions of eATP signaling are to cause fatigue and pain. These are used to signal the need for immobility to prevent further injury. Basically the cells are telling you stop doing what you're doing because it may produce further injury, and to limit transmission of any potential infection, any communicable disease that a person might have that's causing the ATP signaling in the first place. So athletes will, and anyone who's really done, some weight work will know that delayed onset muscle soreness, or DOMS, is a phenomenon of kind of aching that happens for a day to up to 3 days after strength training, and we believe this is related to this chronic ATP signaling. And then the second thing is you need ATP in order to initiate the healing cycle. Okay, all right. Anyway, so the final the conclusion is that multi-system post-infection chronic fatigue syndromes like MECFS, long COVID, and post-Lyme, and post-coccidiomycosis, which is also post-valley fever, and chemotherapy, fragility syndromes have a common denominator, and we see this as an acquired hypersensitivity to ATP-related signaling that is potentially treatable. All these things are connected, but ultimately all these things can be helped to return back to a normal state once you've removed the acute or chronic trigger. And I'll leave it to this last little, this is a little plug that I believe that Salugenesis is the medical dark matter of the 21st century, that abnormalities in Salugenesis affect mitochondria and are present in 90% of chronic illness. And that's it. And there's my my thank you to the Christini Fund, which is really funded 30 years of our research.
Thank you.
Thank you.
Yeah, that was very good talk. And the one part that stood out to me that I hadn't really sunk in from before, I didn't notice it, was the hypersensitivity. I was always wondering, well, why do we get stuck in this when other people can just heal and like they keep going through the healing cycle? So I thought that was really interesting. So what we're going to do is have the panelists who are here, if you have any questions, you can raise your hand and I'll call on people. And then for the attendees, if you have a question, you can type in the Q&A, and Rochelle will keep an eye on those and we'll bring people up as we see questions to add to the discussion. So we will start with Thomas.
Hi, Dr. Naviaux. Fascinating talk as usual. In the discussion webinar chat, we were wondering if you had any thoughts on purerin, this natural product that can be extracted from kudzu, of all things, and seems to have anti-purinergic activity. Could that be a more accessible compound than suramin?
Yeah, so because it's more accessible, we've already been there, done that, tried it in autism, and in a few cases that we've tried it in uncontrolled experiments, it has resulted in not the core symptoms of autism did not improve, and that's all I can say about that. Is that yes, it's out there. But it doesn't seem to have the same overall benefits as a non-selective anti-purinergic agent. So the kudzu, of course, has been the extract has been used in traditional Chinese medicine for probably thousands of years, and it's only in recent years that the compound that you mentioned has been identified. And I believe that most of the studies show that it's mostly focused as an inhibitor of P2X7, which is one of the ionotropic, the potassium and calcium channels activated by ATP signaling. So that's part of so P2X7 is part of the problem, but it's not the only problem.
Thank you. And I should also mention, it would be nice if the people who come up and ask questions could just give like very short, like what's your background? Because we have a very diverse group here from clinicians, researchers. So I just wanted to mention Thomas is on our team, and you just want one sentence. What's your background?
Yeah, I'm a neuroscientist, postdoc at UVM, and I'm also work with Mike Levin at Tufts, who I know is a fan of some of your work.
Okay, thank you. So we'll go to John.
Yeah, so my question, I'm John Houghton. I'm the medical director with Renegade Research, and had a self-history of chemo and radiation a long time ago, and long COVID since then, and so played personally with some of the chronic healing pathways. But in along those lines, of where fibrosis fits in from a timing perspective. So if ATP release starts the cell danger response, then how fibrosis works in a longer time frame to sort of help the body hang out, but also impair function.
Yeah, so fibrosis is what nature uses to provide mechanical support for tissues that have been chronically inflamed and cannot return to their previous biomass through activation of resident stem cells, let's say. Okay, and so the inflammation, can lead to cell dropout if that dropout is not countered by the influx of, or the recruitment of resident stem cells to replace the lost biomass. Then, in order to avoid ruptures in tissues, or then fibroblasts will lay down more, kind of undifferentiated collagen, and that can lead to gliosis in the brain when there's cell dropout in the brain that is associated with, let's say dementia, disorders like Alzheimer. There's also fibrosis, same in lungs. I would say that, the more oxygen in a tissue, the more predisposed it is to fibrosis. So that's another key thing.
Thanks.
But ultimately, we believe that, and I know from some of our, so one of the great immunobiologists that we worked with is Ellen Heber-Katz, formerly of the Wistar Institute, who recognized early in her regeneration studies that fibrosis blocks regeneration. Okay, so it's a problem, but I think it's like in a way it's like senescence. It's a last ditch effort in order to save the system, and that's why we've put that. If you look at our CDR cycle on our web, you'll see fibrosis and senescence both emanating from this, what we call CDR2, the second phase of the recovery cycle, of the healing cycle. Yeah, so key, I don't know how to fix it once it's happened. So I'll leave it at that.
All right, thank you. Rob?
Okay, so I'm Rob Fair. I'm a physiologist. I've been working on MECFS for about 8 years. And Bob Naviaux's theory is one of my favorites. But there are parts of it I don't understand. And so I want to ask one key question here for me. And that is that the purinergic signaling that takes place after release of extracellular ATP has, as you point out, the possibility of starting up ionotropic signaling or cyclic AMP signaling through various of the purinergic receptors. And what I'd like to know is which of those two receptor-operated signaling pathways is responsible for the hypometabolism. How does that come about?
Yeah. Okay. I'll give you. So a lot of our discoveries happen by accident. Okay. We do an experiment in the lab, and we get a result that we didn't expect. Okay. So many of you know that you can purify mitochondria and you can put them in a little chamber. We were using the Ouroboros chamber, but you can do this also in a seahorse analysis. It turns out that if you add extracellular ATP to mitochondria without any cellular, without being, inside a cell, there's instantaneous hyperpolarization and decrease in ATP synthesis, by a kind of a Le Chatelier principle, back pressure across the adenine nucleotide translocator, and an increase in reactive oxygen. That happens instantaneously. But the really interesting thing is that only produced about a 30 to 40% decrease in oxygen consumption when we did that. Yet we had just finished a series of experiments where we had done exactly the same thing, injecting into mice, that ATP, and they dropped their oxygen consumption down to 74, to 26% of what it had previously been. They dropped it by 74%. And so that forced us to imagine a kind of cooperative system where cells were signaling and then likely the CNS is also signaling, and it reminded us of this phenomenon that happens in when an infant sleeps, particularly an infant who has a neurometabolic disorder, a mitochondrial disorder, but all infants do this, is that if you have them you have them in the hospital and just in a bed underneath a plexiglass bubble, and just measure their oxygen consumption and CO2 production. As soon as they go to sleep, within 6 breaths, their oxygen consumption rate, drops by, well, in the cases I can tell you about 60%, A normal child will drop 30% in the first 6 months or so. This gets less and less as they get older and older. But anyway, and then, so it made me think, okay, well, how is that happening? It's happening, virtually instantaneously, but it's happening because of a change in the brain control of peripheral metabolism and all the mitochondria in the body. Is because the only thing that's consuming oxygen and essentially in that sleeping infant or a wakeful infant is their mitochondria in their muscles and nerves, and yet during sleep they instantaneously dropped that oxygen consumption. And so it's the only way that we could put together this discrepancy from the in vitro results, were relatively modest decreases in oxygen consumption in response to direct ATP, and the systemic response from either injection of ATP in the mouse or natural sleep in a child. So ultimately, I don't. To get back to your question of hypometabolism. It is a multifaceted response that I cannot tease out, by inhibiting individual components of the response, we can get partway there, but the whole, the natural response as it works together appears to be very, very efficient CNS regulation of peripheral metabolism. Which gets us back to, brainstem, issues that, could be a problem with CCI. So yeah, I don't know. So I answered your question by, by presenting data that we can't explain. So, but yeah, the answer's in there somewhere. So.
That was great. So I started off this meeting by saying that, I had this dream, and I tweeted that I wanted to be in the same room as Robert and Rob. And so thank you for just fulfilling that. That was great to listen in on. We're going to jump to Nancy next, because she has a hard stop, and she's another one of my scientific heroes. So she gets to skip the line a little bit here.
I always have trouble aligning things.
It's hard to hear you.
Can you hear me better now?
Yeah, that's better.
Oh, great. Okay, thanks. Sorry, it went to my headphones. The how do I align the hypoperfusion data from Dr. Wust's before and after muscle biopsies in long COVID, or from David Systrom's peripheral shunting data, and the mitochondrial work that you have done? Does it fit? Is it parallel?
Yeah,
or
it
is
fits perfectly. I mean, but it comes down to, I mean, you have to, you have to accept a certain perspective about, that is very mitochondria centric. Okay. And that perspective is mitochondria suck. They don't blow. So, so mitochondria set the oxygen gradient of the cell. And if they are not consuming that oxygen, then there is what operationally a shunt. I mean, it basically means that, oxygen that was delivered to the cell is not consumed by the cell. And what you see, in the invasive, cardiopulmonary stress testing, looking at mixed, looking at mixed venous, oxygen, it's the mixed venous oxygen returning to the heart is higher than you expect, meaning that the muscles are you not hearing? Is everyone else able to hear me?
Yeah, I can hear you.
Okay. So because basically, our belief is that mitochondria are sending the signals that determine whether the local capillaries are, whether they dilate or not, or, but ultimately, if the mitochondria, so here's another, Bobism. You can lead a mitochondria into oxygen, but you can't make him breathe. So it's literally, if the mitochondria are receiving signals that tell them to decrease their oxygen consumption and ATP synthesis, they will. And that's what I believe is happening in MECFS, is they're receiving signals to decrease their oxygen consumption.
But could that be because they're not receiving oxygen because of the hypoperfusion state?
Yeah, so so you keep calling it hypoperfusion, but so I believe it's just a physiologic, inability to actually use that oxygen.
Okay.
So yeah, so
that's very helpful. I really appreciate it. And just one little throw out. Am I assuming that colchicine is not something you would consider a reasonable anti-purigenic drug?
right, but colchicine has anti-inflammatory properties, but you get into trouble pretty fast with its limited therapeutic index. So I mean, it's used to treat gout, obviously, but it's not something that it doesn't fit into the antipyrenergic toolkits. It's more related to kind of downstream effects on NLRP3, inflammasome activation and assembly.
Well, thank you very much. I appreciate it. It's nice to see you.
Sure.
Thanks for coming, Nancy. All right, we'll go to Freshta.
Thank you so much. I am also a great fan of this hypothesis and really fascinated about the biology behind that. And by the way, I'm Freshta. I'm a pharmacist and pharmacologist and have been working in the field of precision medicine. And since 2015, working with collaborators, including Ron Davis, Mike Schneiders, and many more on MECFS and related comorbidities. My question for today is the fact that the hypothesis is based on this assumption that the actual insult is gone, and then patients
the what is gone? What is gone? What is gone?
The initial insult is gone, and patients are on trajectory to healing. But then because the recovery is very different from the path that you get actually injury. So maybe the body is stuck to find the best mechanism to get back to where it started, to a healing homeostasis. But what about the heterogeneity in the patient's population and people that actually their journey is in their disease status is when and where they are still having issue with insults such as persistent pathogen, pathogens reactivation, or there are people who actually are among categories that their condition starts from a late onset congenital genetic disorder, like people with lipid disorder, lysosome storage disorder, glycogen storage disorder, all this type of things. So how we could address their issue by trying to help them when the initial insult is still in their body, it's either because they have some genetic issue that some environmental trigger this and they are having MECFS associated condition or symptomology, or the term itself, or they have other things that keeps causing this, the biology being affected. And on the other hand, what about the fact that ceramide itself causes fatigue and some other health related manifestation that actually we do see them in MECFS community? So maybe I would love to ask you, what's your thought on finding the cohort that actually among the patient subset might benefit the best based on where they are in their disease biology and pathology, and find them if they benefit the best for ceramide, and then set up a clinical trial.
So first, I want to correct a misstatement, and that is that ceramide causes fatigue. When you use it at the proper low dose, it does not. Okay,
Oh,
so just as aspirin, if you take 50 tablets of aspirin, you'll have a different effect than 2 tablets of aspirin. Okay, and same thing with ceramide. Ceramide has been used at very high doses. Those are not the relevant doses for MECFS or autism treatment. The next is that so my day job is actually in human genetics and inborn errors of metabolism, and so these disorders that you mentioned are ones that actually we've managed for years, and it's what. And maybe the best example to raise on that is the example of phenylketonuria. So PKU results in a very strong phenotype. It results in from a defect in the phenylalanine hydroxylase gene in most cases. And if that disease is not diagnosed in the first weeks to months of life, and the child is given normal amounts of protein in breast milk or formula, they will develop permanent brain damage that cannot be recovered. Okay. However, if you diagnose early before symptoms and you are able to provide them with a formula that has a decreased amount of phenylalanine and a little bit more supplemented with tyrosine. They go on to have normal brain development, and they're, in very large studies now, their IQ average is about 91 as adults, but there's absolutely no other neurologic or, manifestation of this very strong genetic predisposition that they still have. Okay, they still have the gene. Okay, they still have it, but they're but by able to treat them biochemically, they go on to have a normal life. Okay, yeah, so a lot of diseases like the ones that you mentioned, for example, the lysosomal storage disorders, result in structural abnormalities very early in the process of expression of the gene abnormality. And when there are structural abnormalities, we've always said that there's a problem. I mean, it's difficult to treat those disorders and to take care of them. But I'll bring up one other example. We know through our experimental work with a double-strand RNA simulation of a viral infection that we can give a pregnant mouse an injection of poly-IC, a double-strand RNA, which is completely metabolized and gone within 24 hours. That pregnant mouse will have a mild fever and expression of the CDR, the acute CDR, in the first 24 hours. But her offspring will have permanent activation of microglia for life. Okay, so there's an example where we are 100% certain that the triggering event is gone in those animals. Okay, yet for the life of those animals, they have microglial activation in the brain and inflammation, cognitive abnormalities, behavioral abnormalities that are reminiscent of that are autism-like in many features. So I'd say, yes, there's heterogeneity, and we have to pay attention to the heterogeneity of, that's what good medicine is all about, is recognizing the individuality of every person. But there are going to be precision medicine-based treatments that are relevant to the individual, their personal, situation, and their home. I mean, obviously, you have to pay attention to where they live, how they live, what toxins they're exposed to, occupationally or at home. But there's also, I believe, so this is another thing that came from our 2016 paper. About 80% of the abnormalities that we see in the metabolome of patients with MECFS are personalized, i.e. not shared by the other individuals with MECFS. But 20% are shared. And that 20% of those abnormalities are with the focus of, our work on the cell danger response. That's really what, the other 80% really reflect unique differences on, whether that person's disorder was related to HHV6, EBV, coccidiomycosis, et cetera. They have hallmarks, or long COVID. Now, SARS-CoV-2, is also going to be, will have, in that case, more problems with lung, heart, and kidney disease than an EBV infection, does. So, but I guess, the key is that we have to do both. One, pay attention to the individual and their unique, story and circumstances that led them to where they are, but then also develop, a treatment that that really concentrates on that shared core of abnormalities that could help everyone. Okay, so that's it.
I cannot agree more with you, specifically on the last sentence. Thank you so much. Amazing discussion.
Yeah, thank you. I just wanted to clarify, did I hear correctly that you can, within your metabolome data, you can tell what the cause was? You can separate by the different
No, that's where that those 30 pathways came from. So it turns out that because of the a genetic and biochemical uniqueness of an individual, our disease in terms of metabolism is related to pathways, not molecules in most cases. Okay, and so it's the shared pathways of the cell danger response that were highlighted by the metabolomics work. Okay,
Gotcha. Okay, thank you. Rivka?
Hi, first of all, thank you, Renegade Research, for organizing this, and thank you, Robert Naviaux, for your amazing work. So I'm a longtime ME advocate. I wear many hats. I'm also an advisor to Renegade Research and a PLRC patient rep for the Lisa Saline lab at UMass Chan Medical School and Roshan Kumar's Hi-Fi Biotherapeutics. So my question is about treatments. Sorry, my brain is really foggy today, so if I miss this, please excuse me. But so do we want to boost mitochondria, or wouldn't that just produce more of the extracellular ATP, which I think we don't want? And if we do want to boost mitochondria, have you given any thought to red light therapy that studies have shown boost mitochondria? Thank you.
Yeah, so excellent question. You're not foggy at all. That's a perfect on point, question. Back in the early 2000 aughts, I was an advisor for NASA and a newborn biotech company that was developing near-infrared light therapy for mitochondrial biogenesis and inflammatory disorders, and to promote healing. And what I can say, okay, okay, I'll go back to your first question about more or less mitochondria. So one of the common features of the biopsies that keep coming out is there's a decreased biomass of mitochondria. There's also, so in the mitochondrial medicine world, we typically use citrate synthase, which is a mitochondrial marker that goes down when you have fewer mitochondria and goes up when you have more mitochondria. So citrate synthase is a relatively convenient, rough, measure of mitochondrial biomass, and that is typically decreased in patients with MECFS. Now, what I'll say, what's really surprising, and again, these contrasts. So I'm looking around for Rob Fair. I hope he's still here because it's really a lot of discoveries about the quest for the anomalies. Okay, like you just you just chase after the things you don't understand. And one of the things that we didn't understand is when a child is born with a genetic defect in mitochondria, they proliferate their mitochondria and their citrate synthase often goes up. Okay, that's not what is happening in MECFS. Okay, so it leads me to this conclusion, since I know it's not a shared genetic problem that all these people that are genetically unique are having in their muscle biopsies, that's causing this change in their muscle biopsies, then it has to be a signaling problem. Okay, so what I believe is happening is that the mitochondria are receiving the message to dial back. Okay, and so just increasing the biomass, like if you force an increase in mitochondrial biomass, maybe you'll get some initial benefit, but if you keep pushing that, you'll get to a tip over where there's too much ATP being released from the cell, and it just will produce crashes. So so I worry about that. Like if you focus only on the mitochondria and try to make them do something contrary to their instructions, then you don't solve the disease. Okay, you have to get at the underlying instructions. What is maintaining those instructions that the mitochondria to kind of decrease their overall electron transport activity, or aerobic activity, and specifically fatty acid oxidation, a lot of these other things that are happening. And that's where I'm where I often have problems. So the unique thing about near-infrared light therapy is that it's not specific to mitochondrial biogenesis. It produces effects on scores of pathways that we know now. And I guess I have to say that I have a very open mind for near-infrared light therapies for some disorders. I don't know that it's a game changer for MECFS, but it definitely works in more pleiotropic ways than a molecule that like PGC1 alpha. Like if you really tried to pump mitochondria in a cell, I know from genetic studies in mice that if we overexpress the master mitochondrial biomass regulator PGC1 alpha in heart, we can actually produce congestive heart failure in the mice by having too many mitochondria that are not perfectly integrated with the rest of cell function.
So can I just
Yeah,
can I just summarize what I think you said, which is that it could be so something that boosts your mitochondria could be helpful to a certain level, but after that it could be dangerous.
Well, dangerous. I think that it could. I don't know about dangerous. I would say that it could produce some of the same symptoms that you associate with crashes, for example.
Okay, thank you.
Yeah, that was a great question, Rivka. That was something I was wondering too. We'll go to Jeff next.
Hey, hi, I'm Jeff Wood. I developed the mechanical basis model of MECFS in 2018. And currently I'm on a neurosurgery research team. But I should be clear that all the thoughts that I'm expressing here are just my own. I'm not representing anybody else. So first of all, this talk has been really fascinating. I'm especially interested in thinking more about how a purinergic signaling model might relate to PEM. So it's just really fascinating. I'm sort of having a hard time reconciling one piece of the CDR model with what we're seeing in CCI patients who also have MECFS. And that is that the premise here, as expressed here, seemed to be that CCI would be a result of MECFS, whatever MECFS would be, right? We're positing that we don't know. And just from what we've seen sort of in the patient community, you will see patients develop CCI clearly from a trigger such as an impact trauma, where it's just a clear-cut case of CCI on imaging in terms of symptomology, very, very traditional case of CCI, and they develop MECFS right then and there, So it seems that we're seeing a lot of cases just sort of within the community where the CCI either precedes the MECFS or they occur in tandem, right, with the CCI just
Sure.
as soon as you get CCI, ME results. This other thing that was really interesting was the idea that that MECFS sort of degrades connective tissue through the hydroxyproline and the P5C. That was really interesting, measured in MECFS patients. I'm curious if that has ever been measured in just EDS patients, if there are abnormalities in those values, as they're collagen, they relate to collagen. So curious about that. But really, it's just reconciling sort of the order of things is presented in the very beginning.
So this is how I view things. Okay, it's a circle, and that you can enter the circle via a traumatic injury. You can enter it via a chronic inflammatory process that produces progressive joint laxity and hypermobility. You can enter it, actually independent. You don't have to have CCI in order to have MECFS. Okay, so
yeah, I know. That's clear, but I appreciate the
but the only way you can put it all together is if
feedback,
you see it as a circle. It's not you choose the entrance point. And I know just from clinical medicine that what you say is absolutely a fact. Okay, everything you said is absolutely a fact. It's just that you don't need to have CCI to have MECFS, and you don't need to have and actually the vast majority, probably over 90% of patients that we call EDS with MECFS don't have a genetic cause of their Ehlers-Danlos syndrome. Okay, so that's one of the frustrating things that geneticists have had over the last 10 years is that we, referral and evaluation of a number of patients who do in fact have joint laxity with inflammation, interestingly, and many other symptoms, and chronic pain, abdominal pain is a common thing. But when you look at over 20 different, established collagen and connective tissue related genes that are classic for Ehlers-Danlos, different types, 90% of patients do not have them. So to me, that means it's environmental.
Yeah, environmental.
You can
underlying genetic vulnerability.
Yeah, I guess I always backtrack because being a geneticist, we're all genetic organisms. Okay, so genes are always involved. It's just that sometimes you have many genes interacting with many environmental factors, and usually that's the way it is. And ultimately, we have a phenotype that is indistinguishable from somebody else's that is caused by a completely different thing. Okay. So CCI is important, you know, in the patients who have it, but not everyone has it.
Yeah, I don't think that that was posited here. I just really appreciate the explanation of how it can be a circle and sort of the complexities that you introduced. It's just really interesting.
Yeah, well, thank you for your question. Yeah.
Thank you. So we have Roshan, Janet, and then Athena.
I'm Roshan Kumar. I'm a scientist and immunologist. And we're collaborating with Lisa Selene's lab to look at the adaptive immune response in MECFS and long COVID. And thanks, that was a really great talk. I was interested maybe to follow up on this idea of a circle, and specifically with the immune system. I'm wondering, what's known about like upstream and downstream signals? Like, are there specific immune triggers that can activate the cell danger response? And are there specific inflammatory cytokines that are known that can activate it? And on the other end, what's known about the effect on immune cells? Or is this more like a common response pattern among all cells, and immune cells are just going to react the same way?
Well, I will say that every single cytokine and chemokine we have ever looked at has an antecedent that is purinergic signaling and mitochondrial changes in some cases. So like the way that the cell first responds metabolically to threat, and then it engages and deploys, you know, these various antimicrobial and other defense mechanisms that include the cytokines. So in that circumstance, that's part of why we got to where we did, is because every time I got excited about, you know, IL-6 or CXCL1, or any IL-2, really any inflammatory, or even PGE2, you know, some of the prostaglandins, we kept on, you know, it kept on dumping us back to purinergic signaling as the first responder. I'll refer you, there's a great paper in Cell Metabolism by a group, the first author is Fang, F-A-N-G, on the role of, well, specifically stress, memory, and the adaptive immune response that identified ultimately, T cell regulation and production of xanthine. Okay, well, ultimately it ends up being ATP, but then xanthine that ends up producing anxiety-like behavior in a mouse model, and in large clinical trials of generalized anxiety disorder, xanthine ends up being elevated. So it's just a marker of the previous release of ATP, you know, because xanthine is the oxidized, you know, is oxidized from hypoxanthine, which comes from inosine, which came from adenosine. So yeah, so I'd leave it there. But look up the Fang paper. It's a great paper, Cell Metabolism.
Okay, thanks. So in the case here of the threat, with the threat being an infectious trigger, you're saying that it's the actual infection and cells kind of across the board are sensing that. Is that right? And responding metabolically?
Well, that sure. When there is an actual infection, I mean, so there are a lot of circumstances that the infection has been cleared, and yet the symptoms remain.
Because of that response being triggered?
Yeah, because of metabolic memory and epigenetic changes that happen in response. But you know, ultimately we keep on pushing it back, and what we find is that the cells become, you know, sensitized to progressively smaller and smaller amounts of released ATP. So very small environmental threats can still keep the system in defense mode. where those are completely non-injurious, that are otherwise non-injurious. And we know that kind of from what are the triggers of post-exertional malaise? Well, it's exertion or intense, cognitive activity. It's basically anything that passes. So so in the person's previous life before they were ill, that energy envelope didn't exist, or at least it was much farther, much farther out. And so, but they become hypersensitive. And so very smaller amounts of release will produce the same, intense fatigue that just injecting ATP acutely does. Okay. So, yeah. So, so there's this other problem of interferon and, so the whole cytokine world is important. Okay. I don't mean to, diminish any of these things because they're all part of this highly conserved, system that is, designed ultimately to save us from life-threatening exposures. But if it stays on chronically, it leads to chronic symptoms.
Thank you.
All right, thank you. Ron and Janet?
Ron Davis, MECFS and long COVID researcher at Stanford University. Bob, what do you think is maybe the involvement of some of the herpes viruses? Are they providing signals that affect this whole process?
Yeah. So the herpes viruses are, how should I say, I think helping us, they're leading us down, a path of ancient biology that may ultimately help us crack the problem. So in most cases, reactivation is because of this suppression of TH1 and increase in TH2 lymphocyte function. And so any viruses that were latent can be reactivated under those conditions. And you'll see also an induction of increased amounts of antibody to those different viral products. But so what we've tried to focus on, because of its more ancient coevolutionary history with humans, is the HHV6. So specifically, not herpes simplex virus, but HHV6, which has learned to integrate into telomeres and is present in over 97% of us. Not in all our cells, but on average, about 1 in 50,000 cells of our cells contains, integrated HHV6. And it appears that HHV6 has conferred an advantage to hosts who carry it by being an early alarm system, by responding to the milieu intérieur of threat, and altering the chemistry of the cell in a way that helps it become immune to superinfection by other viruses. And there's a whole area of, kind of at the interface of immunology and metabolism called nutritional immunity that I think is playing a role here. But we don't have that particular. We don't have all the answers to the herpes viruses yet. Other than they've been with us for so many millions of years that it's kind of like my answer to Rob is the answer's in there somewhere. We just have to keep looking.
Thank you.
Yeah. Okay, so we've gotten a lot of good questions in the Q&A, so we're going to start bringing some people up to get them in line. So we'll bring up Sergio and Dina if they want to come up. Rochelle will we'll ask you to. But then we'll go Athena, Bo, and then some of the Q&A people that I brought up. So Athena.
Thanks, Dr. Naviaux, for your great work. I'd like to turn back to the biomechanical piece. I'm an accountant from Canada, and in 2023, my daughter and I built a website from the floor, linking some of the conditions, including inflammation, MCAS, MSIDS, all of the viruses that you've talked about. I wanted to know, as you've built decades of work, has there been a paradigm shift? We see some of the things that influence the epigenome, but we see that symptom onset occurs and doesn't occur for some patients. And the same thing applies with biomechanical issues. For example, we could say, I'm a trauma patient, for example, but I realized very quickly in doing the research and in speaking to other patients that experienced no trauma, that my daughter would ensue with infectious origin. So I want to say, has a paradigm shift occurred for you? And following the science and the research, the second part of that I would say is, do you think the biomechanical piece could be playing an upstream role, taking into account the fact that research has been done showing greater than 80% prevalence of biomechanical issues, not just CCI, but persistent misalignment, for example, of the craniocervical junction, which we know influences many different systems in the body, including respiratory, CNS, just based on the position of C0, C1, C2. So I wanted to get your thoughts on that. And we also know that Hafez did studies many years ago on the fibromyalgia group. So it's not just one group, it's many groups that have shown this prevalence. And we know that there are limitations in imaging, which we're still working on, that are not only not available, but are inconsistent across the neurosurgical group. So I just wanted you to comment on those two pieces, please.
Yeah, well, I mean, what the examples that you raised are, essential components of the cell danger response. So, I mean, so we believe that, the CDR is a way that is, the body uses to respond to traumatic injury, infectious injury, poisons, psychological trauma too. One of the interesting things is we, see an overlap of the same metabolite abnormalities in patients with, psychological traumas, as well as, and in cases of some cases of MECFS. So when you raise the issue about paradigm shift, I mean, we all, like as scientists, what we all hope is that our work takes on a life of its own. Okay. And when that happens, it means that it becomes, embedded in the fabric that all scientists use in order to, to do, more experiments and to develop, cures and offer hope to patients who are suffering. And maybe that's so I mean, so maybe in one sense, I hope that one day someone will tell me, so this is a classic thing for discovery in sciences, tell me not true, not new, not you. Okay. Means that it has taken, the idea is so common that people have forgotten its origins. And that's what I really, hope that this work could do for medicine. So I don't know if that really answered your question. I mean, paradigm shift is, no one ever knows what a paradigm shift is. And from the inside, if you're on the inside, you just don't know. It's really how the world responds and whether it makes a difference. And we don't have the answer to that yet.
Thank you. I guess I would also just raise your thoughts about the global consortium, which Tess and her group have really initiated, in my mind, the opportunity to speak about these biomechanical and other systemic issues. Do you see an opportunity? For example, Dr. Huey has formed a neurosurgical committee with neurologists, neuroradiologists, neurosurgeons, and so on. Do you see an opportunity for patients in the future to work together who have also found things that work? I want to say that this team's initiated that, clinicians, breaking down some of the silos in medicine that operate maybe independently and integrating them with internal medicine physicians, for example, who are treating these conditions, and other clinicians who are treating them. globally, including that piece that I always think of because I'm looking at the cohort and the subcategory of biomechanical issues in my mind.
Yeah. So yes, but it turns out that scientists trained in different silos have sometimes different language that they use to describe the very same things, and they can end up being one person speaking in AM and the other one is speaking in FM, and they really have a hard time coming together. It can, for silos that are far apart, it can take months of interaction, in order to come to a point where the two are actually, speaking the same language again. So yes, that is. Well, if you've looked at our website, it's dominated by silos. If you looked at our, specifically the image of silos and the image of each, how we came to our work on the CDR is by studying a dozen different silos using one lens, using metabolism, okay? And looking at, autism, PTSD, depression, MECFS, post-Lyme, et cetera. So there's a lot that could be done in post-CCI, so pre and post metabolomics after a CCI surgery. There's a lot that could be done to kind of bring together. It's really about building bridges between the silos, and that's what we need to do. It's just that the nature of the way people are trained in medicine and science is, in general, people are trained to isolate single variables. That's not how nature works. Nature doesn't, it doesn't do anything important with one thing. Okay, it really, these are all interacting systems. So yeah, I think, the whole biomechanical issue is absolutely essential, but it's kind of what I've learned in talking to, scientists, for years now, is all the best science is correct. Okay, but how do you bring it together?
Thank you. And just one short question, technical question about the PEM that you talk about in ME patients specifically. Has this been evaluated in supine versus upright position? For example, we know that with, let's call them non-CCI patients that are perhaps persistently misaligned. So let's just call them craniocervical junction syndrome. There are many different names we could go by. We know that body position has effects on the CSF flow in and out of the cranium, and that impacts and plays a role in the homeostasis of the CNS and the oscillations that occur. So has there been some consideration for that in the, let's say, the exercise and some of the measurements of PEM pre and post? Has a position, for example, been considered?
Yeah, to my knowledge, I'm not an expert on that, so I don't know that it has been, but I would ask you, again, to consider the role of the biochemical response of mechanically displaced or distorted cells. Is that ultimately all cellular responses end up being the result of biochemical reactions. Okay. And so yes, certainly biomechanical issues can initiate the process, but the process is propagated by chemistry.
Thank you.
Thank you. So I'll comment on both of those, I think. So for the global consortium, that's sort of what we had in mind. Like, let's bring different people together. So I just wanted to thank everybody who has been showing up to these meetings. I know sometimes it is hard because you have so much to do in your own field, and so to come in and learn about different topics. Thank you. And I totally forgot what I was going to say about the second part, so I'll just bring it up later. But we'll go with Bo.
Thank you. Thank you, Professor Naviaux. Wonderful lecture. And I listened with great interest, and as I consider you the central part, a central part of this MECFS is the dysfunction of the metabolism and the mitochondria. I think we all have that in mind. We know that. and you posed the question, who is giving the underlying instruction to not function properly? And I think that's a very good, who is giving, from where, from whence cometh the instruction not to function? And of course, that's a very interesting. And you've been working with mice, if I understand right. And
by now, actually, we have had more human data than mouse data, but yes, we
okay.
started out with mice. Yeah.
Wonderful, wonderful. Well, with mice, you would probably be able to, if you get the ethical approval, you would be able to test different triggers and even trauma, biomechanical trauma, and of course different infections and maybe even stress. Has that been done in to your knowledge?
Yeah, we've done it. I mean, yeah. So we pushed really hard. I have all sorts of data on multiple cytokine effects. Small doses of LPS, and ultimately we keep getting back to ATP. I can't get any more fundamental than ATP signaling. But I do want to mention there is a point that was raised by the starting question that you had, and that is who is sending the signal. Okay.
Yeah.
And so what I'll say is that there are competing signals that are antagonistic. Okay.
Yeah.
The recovering brain has a system of anti-inflammatory signals that comes from what's called the cholinergic anti-inflammatory pathway, or CAP, that is mediated by so by the vagus nerve, let's say, okay, but by essentially by vagus nerve leaving the brain, it turns out there are also spinal centers that are important for this, but I'll focus on the vagus nerve. The safety signals by the brain are always pulsatile and circadian, always. Okay. Anything constant, anything that's not moving, this gets back to another Bobism, whatever, so the principle of the Serengeti, whatever is not moving on the Serengeti is either dead or soon will be. Okay. So pulsatile signals are part of the healing response of the brain. But it turns out that if the peripheral signals or the peripheral cells that are responding to those, let's say anti-inflammatory cholinergic signals are too engaged in local cell danger signaling, that will overpower the fainter pulsatile signals. So the constant, signals of ATP release in distant tissues, and whether those are, muscle, nerve, heart, liver, kidney, pancreas, thyroid, they're all involved. Okay. So in different people to, more or less extent, but ultimately there's this competition. So this is why we believe that so what is ATP signaling doing? That's playing more of a role at the local level. Okay. It's doing something independent of what the brain is telling it to do. Okay. And so I believe that drugs, non-selective purinergic antagonists like Suramin, will help quiet that peripheral, cell danger response, so that the cells now become responsive to the safety signals coming from the brain. Okay. And so there will be ultimately a fruitful synergy between initially, dialing down that hypersensitivity to ATP signaling, that then will, promote response to anti-inflammatory and also prefrontal prosocial, effects. So ultimately, as as the system fully rights itself, the local cell danger signals will just go into the background and then just, change in their normal rate, as throughout the day according to activity. Okay. And decrease in particular at night when you're sleeping, so that the anti-inflammatory pathways from the brain can have the most effect when you're sleeping. Okay. Anyway, so there's a discord, in disease where the periphery is, got taking care of its own business and essentially unplugged from the, from CNS control, more or less, and obviously don't get too, so physically the unplugging only happens if you break a leg or, sprain an ankle or you tear something. Okay. But there can be chemical, desensitization to long distance signals as well. Anyway, so that's how so I'm always thinking about those two things, danger and safety. Danger and safety, and safety cannot have the upper hand until the danger signaling is brought back to the ground state.
Yeah, thank you. Can I pose another question? I should have told you. I'm from Sweden. We're working. I'm a researcher, medical doctor, and researcher assistant professor at Karolinska Institute. We work at the Brainstem clinics, and we have quite a cohort of patients with MECFS and fibromyalgia, and in my view, they are just very little difference depending on who's actually putting the diagnosis. But anyway, in one of our trials we did recently, we collected CSF fluid from 40 patients with MECFS, and now we're doing the proteomics and metabolomics of these spinal tests. And I would like to have your opinion. What would be the most interesting tests to make from these very rare samples of spinal fluid? Do you have any suggestions on what you would like to test on human?
So the problem is there will be important information in all domains, in the protein domain, in the metabolite domain, in the RNA domain. For me personally, and I'm kind of giving you cutting edge stuff that I haven't talked to anybody about, but I guess I'm going to share it now, is that I would look at the RNA fractions.
Okay.
Actually, Elaine, Moreau is onto some of this, but there's a complex landscape to those RNAs, but they can play a big role in the physiology.
Mm-hmm. Interesting. And one more thing, we have a new ethical approval on on pre and post surgical sampling of patients with cervical radiculopathy, whom may or may not have fibromyalgia or MECFS. And the same question there, what would you like to know, or what would you like to test pre and post such a surgical procedure of these kind of patients?
Yeah, well, I mean, part of this is because I'm so, metabocentric and, I'm a big believer in, metabolomics, which in our parlance is both both the polar and non-polar, molecules, so both lipidomics and metabolomics. I'll say that a bigger part of the signature and metabolism is in the lipid domain. That's an interesting thing. But that's, I guess, so pre and post intervention metabolomics will get you to the pathways. The problem is, this is what I know about complex systems, is you don't ever know a complex system until you understand its response to load. Okay. And that's a hard, so what you're really proposing is unloading the system with a treatment. Okay, so
Yeah.
and that's one part of the dynamic circle. Okay, but the other part of the dynamic circle is what is the load that generates the initial response? You know, that you have to then you can unload. But anyway, loading. Yeah, so I guess that's a bias that a lot of people have. It's just not easy to you know to do that. I mean, clinical trials of loads require a lot of ethical oversight. And in some cases, well, I mean, and medical, they have to, So anyway, I don't want. So I would just say that it's really the response to a stress that differentiates patients with MECFS from baseline. Okay, and that's what's so frustrating for so many doctors. It's not because the patient is sensitive to their whole life, okay, which is unstressed and stressed, okay, and they know what it feels like. Okay, but a physician comes, the person comes to the physician and they're talking coherently and able to, most of the time, stand up, sit down, walk along without, serious or obvious impairment. And then, a physician will come to, a categorically wrong conclusion. Okay. Because they are really not understanding the patient's whole life. Okay.
Okay, thank you. I think you can talk long, but I don't know if these two last questions will fit into this, but you kind of skipped a very interesting PowerPoint picture about the aspects of aging. I don't know if there will be time. And a similar question would be the different cellulogenetic potential of different organs or tissues. I don't know if that we have time with that test.
I mean, I can say, so what we know now from, a lot of multi-omics work, so from a number of different labs, that different organs age at different rates. And your particular rate of kidney aging, can be different than another person's rate of kidney aging, and that person might have a rate of heart aging or a rate of brain aging that is accelerated. So ultimately, all these systems have to work together. But we believe, and you could, if you, just, PubMed Naviaux aging, you'll get my opinion on this, is that incomplete healing is what we call aging. Okay.
Yeah.
I'll just leave it there, incomplete healing. So anyway,
yeah, okay.
That's that.
Thank you so much. Thank you. Wonderful.
Thank you for coming. Let's see. So we're going to go to Dima next.
Oh, hi. 1 second. I wrote this down. Hi, I'm an MECFS patient. Thank you, Dr. Naviaux, for the very interesting talk. Many researchers think that viral persistence might play a part in MECFS or long COVID. And it makes me wonder how sickness behavior response might relate to these conditions, considering how similar the symptoms are with fatigue, sore throat, brain fog. People talk about a flu-like feeling and so on, as well as how tied to infection these conditions are. So I'm wondering whether, wondering whether these conditions are more or less very long sickness behavior responses. Do you think this could fit into your hypothesis at all? And does hypersensitivity to extracellular ATP signaling play any part in sickness behavior that you know of?
Yes, yes, and yes. So I mean, in fact, so some of my PhD work was on a virus that could escape the interferon response and cause diabetes in mice. And so we did a lot of work using double-strand RNA that's now called Ampligen, to activate different limbs of this the stereotype sickness response. Okay, the interferon stereotype sickness response, and it is actually regulated by the acute activation of the cell danger response. So this is stereotype sickness is like if so another so there's another in the 90s there were studies where IV infusions of ATP were given to cancer patients. Okay. And with the hope that it would help with their cachexia, this loss of body mass that occurred with terminal cancers. It didn't. But what it did give them is stereotype sickness behavior and with occasional bouts of air hunger and tightness across the chest with anxiety. So there are some things that are directly related to ATP. There are other things that have to do with interferon, because interferon is probably the easiest way, and well, and IL-1 beta. Yeah, so IL-1 beta and interferon are the kind of two ways to induce stereotype sickness behavior in a healthy person.
Okay.
Yeah, so yes, they are involved. These are all connected. I mean, the reason. So this is all just our biology responding to to stress and these well, I won't go any farther into it, but other than just say yes, stereotype sickness behavior is an intrinsic part of the acute CDR. It's not something that is part of the chronic, other than in individuals who might have, certain other things that can, provide intercurrent disruptions in their baseline health.
Okay, thank you. I have one more question. So I assume that since you're looking for a drug other than Suramin, that you've seen it not work, at least in some patients. So I'm just wondering if you have ideas about why it doesn't work and why another one would work better.
Yeah, unfortunately, so we only did 10 patients, with autism. Half got saline, half got Suramin, and everyone that got Suramin, had improvements, some speaking in sentences for the first time in their lives. When the Suramin, was washed out over the course of 2 months, they returned to baseline. So all I can say is that based on that data and a kind of a binomial estimation, we would estimate that it could be over 60% of patients that could respond in autism. And I just have no data in MECFS since we've never used it in MECFS.
Oh, okay. Okay, thank you very much.
So we're going to go Sergio next and then Sam after that. All right, we'll try going the other way around. Sam first and then Sergio if you're ready after that.
All right. My name is Sam Keating. I'm a PhD student researcher at Texas Tech University Health Sciences Center, focusing on bioinformatics around MECFS. I appreciate a lot of the talk around it's all connected and a lot of the intersections across so many different communities with these mechanisms. And as a systems biologist at heart, I'm curious, sort of how confident are you that you understand the mechanisms that differentiate these overlapped communities? Does this CDR model seem to look like it applies to all of these communities equally, or it's clear cut that it's different in exactly these ways? Or is that sort of framing of thinking helpful towards subtyping and treatment discovery to figure out who actually can be a responder with mechanistic insights?
Yeah, good question. So one of the things to keep remembering is that I'm only studying the things that all the patients share. Okay, I'm not studying the things that make them different. Okay. So in autism, you know, it's commonly said if you've seen one patient with autism, you've seen one patient with autism, you know, so because they're unique. But on the other hand, they have a, you know, core set of symptoms that you can recognize in 100% of patients. And what I'm interested in is studying the thing that affects 100% of these patients. Okay. And I know that because of the, you know, weaknesses of any pharmacologic approach or any physiologic approach, and because of the, you know, natural genetic and biochemical heterogeneity of people, that no drug is going to benefit 100% of the patients. Okay. You know, I'm just enthusiastic that both non-selective and subtype selective, you know, purinergic antagonists, you know, will be effective in a large fraction. But more than anything, I want this to actually become a drug renaissance, so that new drugs, right now, pharmaceutical chemists have no motivation to generate new ATP antagonists. You know, even though there's a fairly large literature out there from animal models, but there's no real incentive. And the companies that have started, you know, to try to, you know, move down this path have been plagued by financial difficulties because of the expense of, you know, doing clinical trials. And this other practical matter about Suramin is that it's 100 years old and out of patent. And so it's very hard to make money as a drug company, you know, manufacturing Suramin. So that's a problem. But if we had new drug candidates based on this idea, so we now have crystal structure of a number of the different receptors, and we know that, you know, actually agonists can bind to, you know, one site that's deeper into the in the pocket, and antagonists are a little more peripheral at the surface. There's lots of structural detail we know from crystallography now, where if there were motivation, you know, pharmaceutical chemists could, you know, make rapid progress in developing drugs. And you know, and what Suramin is, the big problem with Suramin is that it can only be given IV. And, you know, it's got a 2 to 8 week half-life, depending on how long you give it. So those are less desirable pharmacological and pharmacokinetic properties. And if we had a shelf full, I mean, that's what I really would like, is a shelf full of these medicines that we could use in a systematic way to try to answer the questions that you asked in the first place. Are there differences? Will somebody be more responsive to a P2Y2 receptor antagonist than a P2X7 antagonist? And those, you know, excellent and more finely stated questions cannot be answered right now because we do not have the tools to answer them.
And just honing in on that real briefly, I know I faced two sides of that question of the different communities that are overlapped that you addressed, but just within the singular drug that's in trials, not even just the shelf of drugs that we hope to have someday, do you know what the mechanistic sort of responder, non-responder might look like for that? Or what are you missing today? What do you need next?
Well, we need a trial. It's not being done, and it won't be done. It won't be the first one in the, so Pax Medicine, you know, Pax Medica is the company that has picked up the baton for developing Suramin, and they're, you know, they've run into a number of financial bottlenecks. They've succeeded in making high quality Suramin that is, you know, ready for clinical trial. They have to, you know, well, they're planning on getting that, you know, doing the first autism clinical trials next year. But, you know, without additional funding, they won't be able to do an MECFS trial until much later. So it's that we just don't have, you know, any data. I mean, the only data we have is in autism, and then, you know, so we don't have any practical clinical evidence and, you know, about Suramin and MECFS. And we can't have in the US because it's not approved. It's not approved for any human use in the US. It's only approved in Africa for sleeping sickness. Okay. So There's no, it's actually illegal to even import it without FDA approval not to the US.
Wow, fascinating. Sergio, are you able to
Yes.
speak now? Okay.
Yes, I hope you are hearing me fine.
Yeah.
Thank you. Okay, great. Thank you for the great talk. And I would like to explain to you that I have been taking 3 antipyrenetic agents, probenecid, that it is a pannexin 1 inhibitor.
Wait, what? Okay, so
yeah.
So, are you in the U.S. or
No,
are you?
I am from Spain, and I have been taking probenecid. That this is this drug for lowering uric acid, and also I have been taking a brilliant blue FCF, that it's also a pannexin 1 inhibitor, and also I have been taking a lidocaine cream, and for a year and a half. So I know for sure that this combo have been having a antipyrenetic effect, because I have been measuring my IL-1 beta in PBMCs culture with LPS and ATP. And the IL-1 beta has been reduced by 50%. So this is my question. My symptoms during the treatment were very important fatigue, a sense of a very parasympathetic tone, like very deep sleep. The mental fatigue was much worse. So my question to you is, do you think this is because of the immune stimulation effect? Because my NKs are through the roof. I mean, they have always been for the last 20 years very, very low in numbers and also in functionality. Now, after 1 year and a half on antipyrenetic drugs, they are through the roof. And for the first time after 20 years, my line tests, 3 of them, lymphocyte transformation tests, also phages test, PCR phages test, and also antibodies, they have come back negative for the first time, something that even antibiotics for years, all kinds, oral, IVs, everything, Dapsone, the best antibiotics, they didn't achieve. But my clinically, I'm not much better, just a little bit. So this is my question is this because of the immune stimulation effect, or because it is lowering the general synaptic tone, given that ATP is needed for every single synapse?
So, so what I'm hearing is that after a year of, we would consider some of these, like the pannexin channel. Well, so probenecid is not selective for pannexin, but also it regulates a lot of the reabsorption of a lot of chemicals in the kidney as well. But getting past all that, you're saying that there are some biochemical and immunologic tests that have improved, but your symptoms are the same.
Yes, and the effect of the antipyrenetic agents in my severe CFS are a very intense fatigue, a sense of a very high parasympathetic nervous system tone. So the fatigue is much worse. And so I don't know if this has to do with the obvious immune boosting effect, or because the synapses are, like, in general, are being reduced.
Yeah.
So my question is, because I understand that ceramine doesn't cross the BBB very well. So perhaps if the cause of this extreme fatigue was the reduction in the synapses, perhaps ceramine will suit better CFS because it will be acting on the periphery without acting directly on the CNS. So perhaps we could avoid this very aggressive symptom of fatigue.
Yeah, Sergio, you're right on the money. I mean, this is something that we believe that this, inability of CERMIN to cross the blood-brain barrier means that it produces its effect by producing a peripheral signal that then allows the brain to then respond, to a decreased danger signal from the periphery. And that, helps with this anti-inflammatory signaling from, multiple centers, including the vagus in the brain. So we believe that this was a really helpful, although accidental, feature of CERMIN in the autism clinical trials, that it did not actually get to the brain, but it produced profound brain effects.
Yeah.
Okay? And in our very first mouse study with CERMIN, we did, analysis of brain synapse or synaptosomes and looked at the electron microscopy of those synapses and the mitochondria. And despite the fact that CERMIN didn't get into the brain, there were profound improvements in synaptic organization and structure and function in the brain. So this is a key thing, and it's something that pharmacologists often bring up, that CERMIN doesn't cross the blood-brain barrier, and I say, yeah, that reduces your side effects.
Yes. Do you think that perhaps the extreme fatigue and physical and mental fatigue that I experienced with the anticholinergic agents might be due to a reduction in the synaptic tone? Let's put it this way. Because both grows, both probenecid and brilliant blue FCS, both of them grows the blood-brain barrier.
Yeah, so the answer, Sergio, is I don't know about that. Okay, so I know, I have treated lots of patients with probenecid, but having nothing to do with cell danger response. In my internship years, regulating, urate, excretion by the kidney, and a lot of different things. But and they didn't have a brain effect. Probenecid did not have a brain effect on normal people, on people without.
For CFS, it does. There have been like 4 people that I know, and it has a very powerful effect. And it is very similar to taking the lidocaine cream or to taking the brilliant blue FCS. I mean, it is very recognizable.
Yeah, well, it's very interesting. You're teaching me something, so I did not know this, and this is, something, that's very interesting. But I do think that these things, this is telling us that, nature put so there's the control center and the responding centers need to be under different metabolic conditions. Okay, the brain and the body need to be under different conditions in order for the brain to optimize the performance of cells in the body. And if they have the same, I'm prone to digression, Sergio, and so maybe we should, have a beer sometime where,
I love that.
Okay, so, but I think that the insights, your biologic insights are great, but it just reinforces this, kind of complex landscape for drug development that we have, is that if we don't pay attention, we're going to actually make people worse. But if we pay attention and work on, the peripheral, maybe we need to peripheral cell danger response. Maybe that's what I have to talk about. Is the peripheral cell danger response, not, because the CNS is, responds to danger by pulling back on its safety response.
Yeah, interesting. Yeah, interesting, perhaps. Yeah. May I ask a very, very quick question, please?
Yeah, go for it.
Right. Why do you think that when you used 20 milligrams per kilogram of CERMIN in your study of 10 autistic children, it worked really well, but in the other study with more kids, they used 20 kilograms, 20 milligrams per kilogram. It didn't work, but 10 milligrams per kilogram did work. Do you have a theory as to why?
Oh, yeah, and it's called allometric scaling. So in pharmacology, there's it turns out that so a 20 milligram per kilo dose in a 5 or an 8-year-old is equivalent to a 10 milligram per kilo dose in an adult or a 15-year-old. Okay, so it produced, in other words, it produced the same blood levels.
Yeah.
And so it's the blood levels that the patients are responding to, and whether you use 10 or 20 milligrams per kilo to get to that blood level, doesn't matter, except when you overshoot and you can cause more, more side effects.
Okay, so I am understanding from this that your study had younger children.
That's right. We did have, we had some, 2 14-year-olds, but
okay, yeah.
all the rest were young.
So they have a higher metabolism. So that's
yes,
why.
the young,
Okay.
yes,
Okay.
absolutely. And we've actually looked carefully at the Pax Medica data from South Africa, and the 20 milligram per kilo group actually was on average 10 kilograms heavier than
that makes total sense. So perhaps in CFS, we should get closer to the 10 milligram per kilogram.
Yeah, well, I mean, we have created protocols for this, and it's going to be more closer to start off with 5 milligrams per kilo.
Okay, thank you so much for your time, and congratulations for your great work. Thank you.
Thank you.
I'm going to have to get going here pretty soon. We could, take maybe
Go on
1
forever.
or 2 more, but I'm going to have to get going.
Yeah, I did warn you about 3 hours. I'm impressed that you stayed this long, but yeah, it's been going for a while. So we'll go James and John, and then we'll end it there.
Hi, I'm James. I'm from the Davis Lab at Stanford. I've been impressed with your work for a while, Dr. Naviaux, and I had just quick questions. So you were talking about the importance of eATP when it comes to MECFS and these sort of chronic conditions, and that you found heightened sensitivity to it with some things. Do you believe that MECFS patients have increased extracellular ATP or heightened sensitivity? I guess was one thing I was wondering.
Yeah,
And
so not that we can, so we've looked, and so in the vast majority of patients, you cannot see a difference in the plasma levels of ATP.
Okay, so then you'd believe it'd be a sensitivity
Well,
issue?
so this is where you need to get, so if you get it, so turns out cell biology is even more complicated because there's an unstirred water layer of about, say 50 nanometers that surrounds every cell. It's in the, this zone that is occupied by, glycoprotein receptors, the cytokines, and all the different, membrane channels are in this shell around cells that is not responsive to, immediate changes in the plasma. Plasma just moves through. So the way I often think about it is, if you imagine yourself as an angel above a Costa Rican the canopy of a Costa Rican rainforest, okay, you'll be buffeted by the winds, okay. But as you descend into the rainforest, now you smell the scent of the forest, okay, and the winds have gone away, okay. So that's the actual receptor ligand binding zone around every cell can have 100 to 1,000 times higher concentration than the concentration of that same chemical in the blood.
Okay, interesting.
Okay, so for example, ATP in a cell, inside the cytoplasm of a cell is 5 millimolar. Okay, outside in the unstirred water layer, okay, it's 5 micromolar, 1,000 times lower than it was inside the cell. And in you just go another micron away, okay, and go into interstitial fluid or in the blood, it's about 50 nanomolar. Okay, so about 100 times less than it was in the, that pericellular or unstirred water layer of the cell. So it's not actually possible to, answer your question looking at blood, okay? You have to actually look at the cells responding to endogenous and neighboring signals. That's why I keep getting back to the local, how things are locally changed. And that will change cell stiffness that Ron's already measured. It'll change the electrochemical properties that Ron's already measured, and in the MECFS cells. So what ultimately we find is that it's a post-receptor hypersensitivity, so that smaller and smaller amounts of little jets of ATP release will produce greater and greater response.
Okay. Do you have any ideas or like targets for receptors that could be modulating this hypersensitivity?
Yeah, I keep getting it. So yes, so Sermon. Okay, so that's like we keep getting back. There aren't anything. I mean, there are available for preclinical work. You can purchase certain receptor subtype selective drugs that you can test in cell culture and in animal models. Okay. Gamma thio ATP is one that is commonly used, but there are many methylene ATP. There are a lot of different ones that can be used in culture to interrogate different systems. But what I would encourage you to have in cell culture is a load response paradigm, where you're looking at the dynamics of response with and without drug, with and without drug. So, because otherwise you'll get to the situation that, I'm an old gene therapist, and we cured 12 different, genetic disorders in tissue culture, and none of them worked in humans or animals for any long time. And ultimately you just have to remember there's certain questions that you can ask, but the scalability, humans are multicellular, and actually phenotypes are the result of different cell types interacting, not the same cell type. But experiments are done in single cell types usually.
Okay, got it. Thank you.
Sure.
All right, we'll do John is the last question. Oh, you're muted, John.
You're muted, yeah.
There. A quick question, just anecdotally, do you know of any, and then I have the key question, but anecdotally, do you know of any examples where somebody was treated for African sleeping sickness that had MECFS and ended up having an improvement in both? Just
short answer is no. And this is a question you're in very good territory, because Ron Davis asked me that question years and years and years ago. Okay, and it's an excellent question. That's one that I still don't have an answer to.
And then the key thing I wanted to close question-wise was just double checking an understanding of your circle from health to impairment and then back through salugenesis. That from health to impairment, there are multiple layers and steps and pieces that can get you there, whether it's pollution or viruses or trauma or all sorts of things. And is it basically a single mechanistic pathway back through salugenesis? So thinking of it as
okay, so that's really key. Okay, that's a very key. It's because that return pathway is evolutionarily conserved. Evolution couldn't hardwire all the different ways a cell responds to a pollutant that had not even been invented 1,000 years ago. Okay, but it has hardwired how cells actually recover from any infection, a cut, a burn, trauma. Like, it's all the same, only the timing varies.
That's in just double checking, taking it to another system, that when wounds heal, you break down and then clean up and recover, and that's a sort of micro example of a salugenesis. Yeah. Okay.
Yeah, it's perfect. It's exactly right.
Thank you for sharing that single. For me, that single picture on on your slide of the many to one and then a singular pathway back is really helpful for thinking about how do you work the layers of insult as you're getting to recovery.
Okay, thanks.
All right, thank you. I see you're raising your hand, Athena, but we were going to end. Is it like really quick, or did you just have a comment?
10-second question.
Okay.
Because we're speaking about the geographic proximity and some of the issues, when you spoke about infectious testing, do you test for Babesia? Because the numbers are higher than ever.
Yeah.
And you're speaking about some of the medications that overlap. Yeah, that's it.
Yeah, I mean, a lot of these, sort of tick-borne disorders that you get, kind of, Babesia can, can piggyback on a Borrelia infection. And so it's, so, in one sense, the job of the clinician is not made any easier. You still have to go after all the different things that could be triggering the CDR, and Babesia or Bartonella, mycotoxins, a variety of different things, could still be playing a role in, keeping a person, blocked essentially. And and that's being a good doctor requires, thinking of all those different things. But again, I mean, our job is to try to look at helping, the body, once those things are removed, what's blocking the body from returning to health? Okay, that's our big question. And that's where those Salugenesis therapies, and I hope that eventually by the time I die, there will be scores of Salugenesis therapies out there. But at the moment, we only have a few mechanisms to play with.
Thank you.
All right, that's a good place to end it. Thank you again. Thank everybody for your time. People almost everybody stayed around for this full 2.5 hours now. So it's really amazing.
Okay. Thank you very much,
Thank you.
Tess and
Thank
Isabel,
you.
for inviting me, and everybody else for being here and your great questions. It was really a great meeting. I really appreciate it.
Great.
Thank you so
All right.
much.
Have a good day, everyone.
Thank you.
Thanks.
Bye-bye.
It's great.